Hi and welcome to Module 8, our last unit of our Year 12 course. And in this module, we are looking at chemical ideas. So far we have studied a whole range of things such as quantitative analysis. We've looked at organic chemistry and their structures. We've looked at equilibrium and acid-base reactions. And in our Year 11 course, a whole heap of things: gravimetric analysis, volumetric analysis, and this last unit is that unit that ties all these things together. So the identification and the analysis of chemicals is of utmost importance in the scientific community, particularly medicine, environmental management, quality control, mining, and so forth. We're going to be looking at a range of methods that chemists use to identify and measure quantities of these chemicals. So you're going to be looking at investigating and processing the data from a whole range of different tests, such as testing ions in solution and determining what ions are present there or any there that could be of concern to human health and the environment. So with our water quality, are there heavy metals there? Are there high levels of metals that could be dangerous to our health, and so on? You're also going to be looking at organic structures and being able to identify and confirm the structures of some of those organic compounds from using a range of data such as chemical reactivity, but then also interpretation of some of the other tests that we might perform, such as infrared spectroscopy, NMR spectroscopy, both proton and carbon, and also mass spectrometry. Some of these you've seen already from our visit to the TAFE, and we're going to look at them all a little bit in greater detail. Now we're going to start with our inquiry question 2 at the moment, and the reason we're going to start with inquiry question 2 is it really ties in with the organic chemistry that we have just done, and also ties in with some of the things that we have seen in our TAFE visit as well. So, inquiry question 2, how is information about the reactivity and the structure of organic compounds obtained? So both qualitative and quantitative tests have led to a big understanding of the chemistry of organic compounds, and things such as our mass spectrometry and the infrared spectroscopy have really enabled a deeper understanding of the different functional groups that might be present, as well as their molecular structure. So this module is going to investigate both of these quantitative and qualitative tests to see what functional groups are present. So to start with, we are going to look at chemical tests for functional groups. And I'll just move this down. So the outcome that we're looking at: conduct qualitative investigations to test for the presence in organic molecules of the following functional groups. So our carbon-carbon double bonds, hydroxyl groups, and carboxylic acids. Now, none of these are new to you. You've done all of these tests in our previous organic unit, so we're going to have a look at them again, but this is really just to recall and make sure you understand, linking in with all these other methods that we've got, how important these tests are in providing us some information about the structure of the organic compound that we've got. So we do depend on organic molecules in all aspects of our life: the food we eat, the medicines we use to fight diseases, fossil fuels, biofuels that we can use for our energy production, and even our body is made up of organic chemicals. So chemists are constantly finding new organic compounds and identifying them, but also synthesizing some new ones. And so to be able to do this, they need to know what structures are present and how they react. So the tests that we're going to have a look at are going to be those tests that we have seen before. So we'll start off with our carbon-carbon double bond. So we know there that our carbon-carbon double bond are present in alkenes. So the presence of this double bond can actually be detected using bromine water. So this is simply just bromine and water mixed together. Now, bromine has a characteristic reddy, orangey kind of colour, and when we add an alkene, it will go clear. So if it stays the same, it's not an alkene. If it does, then we know we have an alkene on our hands. So when this happens, it's an example of an addition reaction. And we can see here in our diagram that we have got a double bond initially in our alkene, and that double bond is then broken and a bromine atom is added to each of those, each of the carbons that are on either side of that double bond there. So bromine is red, and when it does attach, it's an addition reaction. Only one product is formed, and we get our colourless solution on the other side. So looking at a diagram here, you can see here in this left diagram over this side, we have got that distinctive orange-red colour, and this one here has turned a nice colourless colour, which indicates that an alkene is present. Now, a disclaimer, it won't tell you whether it is an isomer of that. So if we had butene, we wouldn't know if it was 1-butene or 2-butene. All it is telling us is that there is a double bond that is present. So another type of test that we're going to look at is the presence of a carboxyl group. So our carboxyl group is that COOH that's present in our carboxylic acids such as ethanoic acid and so on that we were using when we were making esters. So they're weak acids. Now, a characteristic property that they have is being an acid, they will react with carbonate salts. So if we react it with a carbonate salt, we should get that typical carbonate reaction and the production of carbon dioxide gas. Now, a test for carbon dioxide gas is piping it through or blowing it through lime water, and if our lime water turns from clear to milky, we know that carbon dioxide has in fact been produced. And because it also is an acid, another test that we can do just to confirm that it is in fact an acid is a test with blue litmus. And if we add blue litmus turns red in an acid. And our other test that we are going to look at is going to be our hydroxyl group. Now, our hydroxyl group is present in our alcohols. So if we've got a dry sample of our alcohol, so that is no water is present, and we add it with a small piece of sodium metal, the alcohol will react violently if water is present, so we don't want it to be there. We will get bubbles of hydrogen gas being produced. Now, we can't do this in the lab, obviously, because it's very dangerous, and if there is any water present, the reaction could be quite vigorous. So instead, I would suggest to you to type in this address into your browser and have a quick look at this reaction of alcohols. And there's a couple of different alcohols there, so you'll notice a difference in their reactivity as that metal is added. But it's a good way to see how that can be tested with the sodium metal. Now, as you know, we've done a whole heap of tests with alcohols with our primary, secondary and tertiary alcohols there, and you can see in this picture that when we actually have our primary, secondary and tertiary alcohols added to our potassium permanganate, which is a nice purple colour due to the manganate 2+ ions, when we add a primary, it goes clear. When we add a secondary, it goes clear, but our tertiaries do not react. Now, with our primary will oxidize to an aldehyde. If there is excess of our permanganate, it will further oxidize to carboxylic acid. With our secondary, it will oxidize to a ketone, but our tertiary won't oxidize at all. Now, a little bit of a summary of some tests here. Another test we can do is add some glacial acetic acid with a little bit of concentrated sulfuric acid, and we're going to be looking at our ester, and we'll get that fruity smell. But here is a little simple test that we can see whether an alcohol is primary, secondary or tertiary based on the reactions that we have got here in the table. So here's another little table, just a little bit of a summary showing some of these other reactions. We've got alkanes. If we put those in bromine water, no double bond, no reaction. It's not going to react with sodium either, and no reaction with our sodium carbonate. If we've got our alkenes, we put it in bromine water, there will be a decolorization, no reaction with sodium, and no reaction with the sodium carbonate. Alcohols, no reaction in bromine water. Bromine water can actually act as a weak oxidizing agent, and it may actually react, but it might take a very, very long time. So in lab conditions, it doesn't, but no reaction there anyway. But if I put an alcohol with some sodium, we'll get effervescence, but no reaction with the sodium carbonate. And an alkanoic acid, no reaction in bromine water, effervescence again with sodium. So similar test there with our sodium, but the difference then is the alkanoic acid, being a weak acid, will react with that sodium carbonate. So make some notes, tie all of these things together, and we will practice a few of those. But here's a question for you that I want you to have a go at, and I want you to bring to class in our next lesson and answer. So, an unknown colourless liquid is known to either be an alkanol or an alkanoic acid. So the following tests were performed, and you need to use this information and justify your answer. So you need to give reasons for your answer to support what you're saying. Test 1, we're adding some sodium metal and it's being added to the unknown and effervescence occurred. In test tube 2, that sample was there, we added bromine water to it and it did not decolorize. And in test tube 3, we added some sodium carbonate to the unknown test tube and effervescence occurred. What type of organic structure does that unknown have? Making sure that you justify your answers. We'll talk then. See you later.